Assalamu alaikum guys. Today I will continue the explain that I did about preparing tissues again. In this explanation, when the tissue first, how is tissue prepared by using pictures? When a tissue reaches the lab, the tissue is put into cassettes and then it's fixed by formalin or formaldehyde, 1% or 5% formaldehyde. This are what the cassettes look like. The tissue is added. Uh, the tissue is put in here. Yeah. Sorry. The tissue is put in here. As you see, I will change the colors. The tissue is put in here, and it's closed. Okay. So the tissue is put in here and it's closed. So after that, you put it in formalin, fix it, sorry. And after putting it in formalin, the tissue is dehydrated by using alcohols in different concentrations. First 50% alcohol, and after that we get out, of course, putting it in alcohol, and we put it in 90%, and we put it in absolute alcohol. What do you mean by absolute alcohol? It means 100% alcohol, and again in absolute alcohol. Okay, after that we will clear it using xylene. Okay, that's an organic reagent. Embedding is simply done by tissue embedding, the technique of placing cells or tissue in supporting medium. This supporting medium so the thin sections can be cut using a microtome. The medium can be paraffin wax. Okay, so paraffin wax is used and put in special mold and after that sectioning is done. As you see here, it's put in a machine called a microtome, and there's a wheel at the right. You spin the wheel. After spinning the wheel, this holder gets up and down. After getting up and down, okay, you will get those kinds of sections, very thin sections. Those thin sections are about the thickness of it is about 4 to 10 microns. Okay, that's how I explain it now. And after that, you would put it in a water bath. Water bath is used to, make the tissues more smooth and just like calming down. The water bath temperature is about 40 to 50 degrees Celsius. It's put in this. It may be just like a pool. There is no difference between the shapes, but the function is the same. And after that, drying is done and it's stained. So we have two main stains: H, uh, H. This is the H. Sorry, H and E. And this is distilled water, okay, for washing the slide. After that, okay, hematoxylin, we put it in hematoxylin to stain the nuclei, okay, and we put it in ozone to stain the cytoplasm. After that, this distilled water is added. After adding distilled water, we will end our process and we dry it and we mount it by a cover slip. Okay, we use a DPX medium, or we can say DPX. It's a special for mounting. After that, we would observe it under the microscope and we will get the tissue.